Welcome back. Welcome back from the short break. I now have the extreme honor of introducing our next keynote speaker, Professor Harry Orr from University of Minnesota. Professor Harry Orr has performed pioneering work in the field of CAG triplet repeat disorders. His presence here really shows how privileged we are in this field to have such brilliant minds working with us. Professor Harry Orr has cloned the gene for spinocerebellar ataxia type 1, together with his colleague Huda Zogby. He has then performed continued work on spinocerebellar ataxia. 2 years ago, he received the very prestigious Kavli Prize in Neuroscience, together with Huda Zogby, Professor Jean-Louis Mandel, that we heard from yesterday, and Professor Christopher Walsh, for their pioneering work identifying genes that are really underlying really severe neurodegenerative disorders. We're very happy to have Harry Orr here today, and we're very much looking forward to your talk.
Thank you very much, Asa, for that very kind introduction. Coming from Minnesota, we're very proud of our Scandinavian heritage, and it's an extreme honor to be introduced by a Scandinavian. Thank you, and you did much better job in introducing me than the individual who introduced me to you yesterday. But in recognition of yesterday's discussion, I changed the title of my talk slightly to Building Bridges Between CAG Triplet Glutamine Repeat Disorders, in recognition of yesterday's event and that we are in Strasbourg. And I fully appreciate that bridges are architectural features now. One more point about yesterday. As a molecular geneticist, I'm very proud that I knew TAG was a stop codon. And as somebody who grew up in the 1960s, I recognized Woody Guthrie's picture. Okay, these are the disclosures. You can read them quickly. There's nothing really to disclose. Okay, so the basic tenet of what has prompted a lot of the studies I want to talk to you today about is the idea that if we understand the pathogenic similarities and differences between, say, Huntington's and another polyglutamine disease, SCA1, that will help us focus our attention on key targets that are likely for effective treatments as we move towards the overarching goal of going from gene identification to developing such therapies. So let me give you a little bit of a background about the overview of SCA1. Like Huntington's, it's an autosomal dominant gene, a disease, the gene for which is located on the short arm of chromosome 6, in linkage with the human immunohistocompatibility complex, HLA. Now, for some of the younger people in the audience, let me just give you a little history here. I got my PhD in neuroscience, at which time my advisor, Oliver Lowry, the protein Lowry, told me, you know, Harry, you need to learn these modern molecular genetic techniques. So off to Boston and Harvard in Jack Strominger's lab, I went for a postdoc and did molecular genetics on the HLA complex. And I was hired as an immunologist at the University of Minnesota and continued to do work on HLA, when one day a neurologist walked into my office. His name is Larry Scott. He is a member of a large SCA1 family, and they had just shown that the gene in his family was linked to the HLA complex. And he said, you know, Harry, I think I have something here that might be of interest to you. Well, so my message to the younger people is keep the doors open, because you never know when something cool is going to walk into your office or your life. So let's see. And the other thing is, a long time ago, God, almost a century, anticipation was identified by another member of this family, a neurologist also, Jonathan Scutt, in 1951, i.e. the disease got progressively worse as it moved from generation to generation. So when we had mapped the region of SCA and we're beginning to now try to go down to the specific gene, we had a feeling it was an unstable trinucleotide repeat because of the work that was being done in Fragile X, SBMA, and others. And we heard through the proverbial grapevine that the HD gene had been cloned and the mutation was an unstable CAG repeat. So the first thing I need to do is thank the HD community for giving us the key insight that allowed us in literally weeks to identify the SCA1 gene, because the first thing we probed with was a CAG repeat. So the disease is typically adult onset. It's characterized by gait and limb ataxia, and this individual here is about mid-course in their disease. And a very classic feature of cerebellar ataxia is this wide stance that you're seeing as he's trying to walk down the hall, and issues with balance is depicted by his hand up against the wall. But in addition, as the disease progresses, you see a decline in executive function, particularly in cognitive issues. There's muscle wasting and weakness, difficulties in chewing, swallowing, and breathing, which are thought to be the underlying basis for the lethality of this disorder. The human gene consists of 9 exons, and it's the last 2 exons that contain the coding information, exons 8 and 9. And like we've heard with Huntington's, unaffected alleles range up to around 39 repeats, and mutant alleles are 40 repeats and greater. Like Huntington's, there are juvenile forms of this disease with repeats in the 70 to 80 range. The protein, I think one of the nicest things about working on SCA1 is the protein's only 800 amino acids, roughly about a quarter of the size of Huntington, which makes doing biochemistry a little easier. So we were able to be successful. The polyglutamine stretch is located towards the amino terminus. There is a structural region called the AXH domain, which was really important in the interaction of this protein with other proteins. And then, as we'll see, some key other functional motifs is a phosphorylation site at serine 776 and a nuclear localization sequence a little bit upstream from this phosphorylation site. So, if we now look, what was known is that there's dramatic atrophy in patients with SCA1 in the cerebellum, is shown by this MRI image of an individual at about the same disease state as we see here. And the atrophy of the cerebellum is indicated by the decrease in the fovea and the subsequent interdigitation of cerebral spinal fluid between the fovea, and there is also atrophy in the brainstem. But interestingly, when we looked at postmortem samples, there's a dramatic loss of Purkinje cells. And this is a very prominent and consistent feature of this disorder. So when we first started out trying to understand the molecular basis of this disease, we focused on a mouse model where we, using transgenic approaches, specifically expressed a mutant human cDNA for ataxin-1 with 82 glutamines in Purkinje cells of the cerebellar cortex of the mouse. Now, it's important to note that in this model, we were overexpressing the ataxin-1 protein roughly 20 times that of wild type. So, in these mice, through a progression of a series of experiments and papers outlined here, we came up with a molecular pathway which we think nicely defined the pathogenic process in Purkinje cells. And it's highlighted by three features. One is the phosphorylation of ataxin 1 at serine 776, which is important for stabilizing the protein such that it's not degraded. In absence of this phosphorylation, the protein is very rapidly degraded in Purkinje cells. It also finds a binding site for chaperone 1433, which we think is important in this stabilization. The other feature we found is the transport of the ataxin 1 protein into the nucleus was critical for pathogenesis. And finally, once in the Purkinje cell nuclei, interaction with a transcriptional regulator, Capicua, a transcriptional processor, was also critical for pathogenesis. And here's the sort of study we did with the phosphorylation. If you change the serine to an alanine, which can't be phosphorylated, as we've heard in other discussions, the mutant protein is rapidly degraded, and the mice show no signs of disease. Likewise, if you mutate the NLS, it's a classic monopartate nuclear localization sequence consisting of 4 largely basic amino acids. And the classic mutation for this type of nuclear localization sequence is to take the second lysine and mutate it to a threonine. So in this case, it's K772T. And this shows that this form of protein, unlike the wild type or the normal mutant protein, shows no localization to Purkinje cells. So this NLS is absolutely required for entry of this protein into the nuclei of Purkinje cells. And you see a complete restoration of neuronal function, neuronal phenotype, motor phenotype, as shown by the rotarod data. And this is the same sort of data in terms of restoration of phenotype that we saw in the other mice. And lastly, if we mutate the AXH region, changing a valine at position 591 to an alanine and a serine at 602 to an aspartic acid, which completely disrupts the interaction of ataxin 1 with Capicua, we also see a complete restoration of normal function, no signs of motor phenotype or Purkinje cell disease. So this was sort of our Purkinje cell pathway. But ataxin 1 is widely expressed. And I think a couple of interesting points that will come up later is that it's highly expressed both in the cerebellum and the caudate. So what are the roles of these other regions in the various phenotypes? And is this Purkinje cell pathway, how faithfully does it replicate to other sites of pathogenesis? So to explore this, the next thing we did was to generate a knock-in model of SCA1. And this was done in Huda's lab. And first of all, the mouse ataxin 1 gene has 8 exons, in contrast to the 9. Exon 2 is the mouse gene does not have the exon 2 that we see in the human gene. And so what Huda's lab did was insert a stretch of DNA containing 154 glutamines into exon 7 of the mouse gene to now give us a knock-in version of this mouse, of this disease. And like our Purkinje cell version, we saw a motor deficit on the rotarod, but we also see, unlike our Purkinje cell, we see a decrease in survival. We see alterations in cognitive phenotypes. In this case, it's the Morris-Water maze, and we see a failure to gain weight. Now, you'll note in these knock-in animals this severe kyphosis. And just to give you a heads up, we know this is due to a direct effect of the mutant protein on skeletal muscle. So if we delete the mutant protein from the skeletal muscle, the kyphosis goes away. Okay? So there's a direct pathogenic effect of the mutant protein on skeletal muscle. So, what about our pathogenic pathway? So, taking these knock-in mice and using CRISPR to introduce the same mutations, serine 776 to an alanine, we see an improved motor performance, we see a roughly 20% increase in survival, but no improvement in cognitive deficit or weight gain. If we move to the AXH mutation, we see a similar change in phenotype by making this mutation. Improved motor performance, some increase in survival, but again, no effect on cognitive deficits or weight gain. In contrast, if we make the NLS mutation, in addition to motor performance improvements, we see almost a doubling in lifespan in these animals, a 90% improvement, and this is the largest improvement in survival that we've seen in any manipulation that we've done to our SCA1 mice. We also see some improvement in cognitive deficits and some improvement in the failure to gain weight, but it's not a complete restoration to the wild-type phenotypes. But at this point, and it's still true, the most substantial contributor to the pathogenesis is the entry of the mutant protein into the nucleus. And I would just like to highlight here that we're making a single amino acid change in the protein. And I think this really emphasizes, at least in our SCA1 models, the importance of the protein in pathogenesis. And that doesn't say other things don't contribute, but clearly the protein plays a very critical role in this model of the disease. Okay, now we want to start to begin to understand what's going on in different regions of the brain and body that's affected by this disease. So now this is a second generation of the knock-in model, and basically in collaboration with a colleague at Minnesota, Mike Koob, what he basically did was replace the mouse exons 7 and 8 with the human exons 8 and 9 containing 146 CAG repeats or glutamate. So we sort of semi-humanized one allele in the mouse with a mutant form of ataxin-1, of the ataxin-1 gene with 146 glutamates. And importantly, when we generated these mice, LOXN recombination sites were flanking the two coding exons so that when we cross these mice with CRE, as we'll see in a moment, it gives us an opportunity to both assess the localization of the SCA1 phenotypes from an anatomical perspective, and it also gives us a preclinical platform for screening therapies that are targeting the human gene. So ASOs and so forth and so forth. So here's the sort of quick summary that, that there, in these mice, there's two alleles of ataxin-1 gene, one containing 2 glutamines, encoding 2 glutamines, and the other, the knock-in version, encoding 146. In the presence of CRE recombinase, this coding region is deleted such that now you only have the wild-type allele present in the respective cells or tissue regions. And so now, this is the first piece of evidence that really started to get me to focus on something other than the cerebellum in terms of SCA1 and the brain. And this is a study that comes from Ricardo Pinto and Vanessa Wheeler that we collaborated with. And basically, this is looking at the repeat somatic instability in SCA1 brain. And this is their tracings. Here's the expansion index that they had calculated for various regions. And to me, the really intriguing point is if we compare the ataxin-1 CAG expansion index with the Huntington expansion index, I don't think you really need statistics to realize it's a straight line. And here's the caudate data. So again, very similar, at least from this analysis of somatic instability in the SCA1 patients as we see in Huntington's patients. So let's go to our mouse, back to our mouse. And the critical CRE line is the ROSA CRE, the RGS9 CRE. And if we cross it with a reporter mouse, the LAC-Z reporter, we see up here in this image very selective recombination in the striatum and specifically the median-spiny neurons of the striatum. So we're able to delete now our mutant allele from the median-spiny neurons. And again, in collaboration with Vanessa Wheeler, in a paper recently published in JCI Insight, here's the somatic instability that we see in our 146 knock-in mice at 35 weeks. You can see very dramatic expansion in the striatum, but also in other regions. And there is expansion in peripheral tissue, instability, if you will, at least in terms of the tail. But in our RGS-9 mice, where we've deleted now from the median-spiny neurons, as shown over here, there's a dramatic and significant reduction in the expansion index, specifically in the striatum, but not in other regions and other tissues in this mice. So like Huntington, there is a substantial somatic instability of the CAG repeat in median-spiny neurons of the striatum in these mice. And I just got a sense that maybe Sarah has a question. Okay. And now with Christopher Pearson, we begin to look at the age effect of this somatic expansion. And basically, what you're looking at here is a 20-week-old animal, different tissues. And here we move to 30 weeks and 52 weeks. And if we move down here to the striatum, we see there is this very consistent, it's very similar to what one would see in a corresponding Huntington mouse model or even Huntington tissue. In collaboration again with Ricardo Pinto, he has injected a virus containing that is able to knock out the DNA repair gene MLH3 into these 146 mice, and the absence now of that dramatically reduces the somatic instability both in striatum as well as in the peripheral tissue liver. So again, a very, shall we say, similar situation as what has been shown by many in the case of Huntington's disease. Oops, got to move here, Harry. All right. So with William Yang, we have done a comparative analysis of the differential gene expression pattern seen in our knock-in mice. In this case, we're comparing the ataxin-1 175 knock-in mice, which is a derivative of the 154 knock-in mouse that I had shown you earlier, with several Huntington's, both the Q175 and the Q140. And basically what we're looking at here is if we compare the differential genes between the ataxin-1 knock-in mice versus wild type, we see a very high concordance with the differential genes in these different HD models. If we now do our NLS mutation, so we correct for those genes that nuclear entry is required for, it goes up even higher. And this is further emphasized by if we now compare the KT mutation with the mutant knock-in, there's a complete absence of overlap with Huntington's. So again, it suggests that this group of differential gene expressions that's going on in our SEA1 mice is again very similar to what you all have seen in Huntington's. So, what a little specifically, we've looked at some by qPCR, some key, both in direct, indirect MSNs and interneurons. So for example, here's DR1, DR2, and you can see a dramatic effect going from 18 weeks to 35 weeks. Here are the significant levels. So again, very high level of significance. And these are very similar to what you see in the Huntington model. And interestingly, if we now go to our RGS-9 mice and delete the mutant gene from MSNs, these alteration in gene, we see substantial improvement, if you will, or normalization of the differential expression of these genes with deletion of the mutant allele from median-spiny neurons. In contrast, if we do a similar analysis in our knock-in mice and compare it to the KT mutant mice where we disrupt nuclear localization, we do not see a correction in the differential expression of these genes, which are thought to be key markers of striatal function, MSN function. So alteration of nuclear localization by this mutation in the striatum has nowhere near the corrective effect that we see in Purkinje cells in the cerebellum. And this is further highlighted by, here's sort of a summary of the data from the cerebellar. There's this magenta WGCNA module that is correlated with ataxia in the Purkinje cell model. In the knock-in model, 6 of 10 of the top 10 hub genes in this module are significantly downregulated in the knock-in mice, and 5 of these 6 are corrected by the KT mutation. And overall differential gene expression, 87% of the differentially expressed genes in the cerebellum are corrected by the KT mutation. Okay. So we're getting close here to the end. Lastly, we're moving. It's nice to look at differential gene expression, but I sort of feel it's important to also look at the protein, because that's presumably what's going to drive the phenotype. And a postdoc in the lab, while she was working in Pascal Kassler's lab at Harvard, characterized the 2 antibodies to DR1 receptor and DR2 in basically, here's a wild-type mouse staining pattern. The purple are the receptors. And now if you do a similar experiment where you've knocked out either DR1 or DR2 by using Cree and Aflox mice, you can see loss of staining in the case of DR1 knockout with the DR1 antibody, and similarly with the D2 antibody. So showing specificity of these antibodies for the respective receptors. And if we now go to our ataxin-1 mice, you hear the images, but if we just zip over here to the data, this is a direct pathway. So this would be D1R going down. You can see a significant reduction in the 146 mice, which is corrected again by the RGS9 deletion, and also in DR2. But we're kind of intrigued that the idea that this DR1 may be more sensitive in terms of its change than DR2, and we need to do more mice more times to further substantiate that. I guess, I don't know if Ed is here, but I said I don't work on HD. Well, guess what? We are. Here's a similar analysis in the Q175 mice, and basically we see a similar decrease in both. For some reason, the significant dots disappeared on this slide. So they're both significant decreases, very similar to what we see in our SCA1 model. But again, we're kind of intrigued where DR1 may be more substantially affected than D2R. And again, lastly, doing the similar experiment now in our knock-in model. And again, we see, in this case, D1R this is at 5 weeks of age, so earlier. We see again a much more dramatic effect on the DR1 expression in this model at 5 weeks, essentially no change in D2R. And as suspected from the differential gene expression pattern, the NLS mutation does not correct this. So we're now in the process of doing more of this immunohistochemistry, but also assessing spiny neuron function by electrophysiology. And this is another message to the young investigators. I have sworn for roughly 35 years that I would never do electrophysiology. So be careful what you promise. And so the last 2 slides. In SCA1, at disease onset, we sort of focused on the pathology in the cerebellum and perhaps the medulla and the pons. And then perhaps as the disease progresses, we begin to see pathology in the striatum. And this is what the human imaging data says. In contrast, in HD, at onset, there's very prominent pathology in the striatum. But yet, at least in our mouse models, the pattern of instability in differential expression is identical. So what's the basis for this differential pathogenesis? And I'm really intrigued, and I think understanding this is going to be important for understanding both diseases. So we're open to any suggestions or help. And that's the first take-home message, I think. And the second take-home message is kind of outlined in this slide. And it relates to the fact that the NLS mutation, K772 in ataxin-1, is very effective in correcting the pathology in the cerebellum, but not so in the striatum, in our mouse models, which raises the possibility that the pathogenic pathways vary between affected regions. So while the NLS and the nuclear localization is clearly a prominent driver, it's not, apparently, at least in our SEA1 models, not the answer to everything that's going on in these mice. So here's the group. As mentioned in the introduction, this is a long-standing collaboration with Huda Zogby's lab at Baylor. I've put 1987 in quotes. This is a classic male-female relationship where the male doesn't quite remember when it started. So somewhere around 1987. And the other thing I would like to do is thank the HD colleagues that I've worked with. You guys you're very strong, and you've been very supportive and very interactive. And so it's been a lot of fun. And also, like many of us have done, and not to forget the respective families for your ongoing support. And with that, I will end with questions. I think according to this, we're a minute over. So what does that mean? No questions?
No questions, but we want to give you a big applause. Thank you very much.
I don't know, is Ed here? I wanted you to notice I pointed to the screen every time. I didn't point to the monitor, so I'm learning.
So thank you very much, Harry. Fantastic talk, very thought-provoking. And with that, I leave it over to the short communications.